During ASH 2020, we had the opportunity to hear three very interesting studies on COVID-19 and the hematologic aspects of the pandemic. The first one studied thrombotic microangiopathy variants that were independently associated with critical disease in COVID-19 patients. The second one reported outcomes of patients with hematologic malignancies and COVID-19 infection as a report from the ASH Research Collaborative Data Hub. And the third one reported the use of allogenic off-the-shelf SARS-CoV-2 specific T cells to treat high-risk patients with COVID-19. During the ASH 2020 virtual meeting, we were very happy to announce our results on our COVID-19 study. This was based on our hypothesis that complement-related germline mutations would be evident in patients with critical COVID-19 disease. To test this hypothesis, we used a custom complement-related gene panel, including the gene of Adams TS13 that is also associated in thrombotic microangiopathies, and we ran next-generation sequencing on our samples. We studied 60 COVID-19 patients, 20 with critical and 40 with moderate disease. We found for the first time rare and pathogenic variants in a few of our patients. And also we were able to identify a combination of variants that was evident in the majority of patients with critical disease and in a very few patients with moderate disease. In the statistical analysis, this combination was independently associated with critical disease. This finding is a very important one and is going to help us identify patients that would benefit from complement inhibition if we can validate it in a rigorous algorithm and artificial intelligence model. And of course, if these results are verified by the phase 2 and 3 randomized trials of complement inhibitors. During ASH 2020, we had the opportunity to learn more about COVID-19 and hematologic malignancies through the ASH Research Collaborative Data Hub. And this is a very interesting data hub, including 250 patients with blood cancers from 74 sites around the world. These were patients with hematologic malignancies. The majority of them had acute leukemia. And these patients presented several syndromes and were treated according to different protocols. What is very interesting from this study is that patients with hematologic malignancies experience significant morbidity and mortality from COVID-19 infection. Of course, we need more data to further understand which patients and which disease is of greater risk, and if we can do something to improve the treatment of these patients. And therefore, we encourage all our colleagues around the world to submit their cases in this collaborative data hub. During the ASH 2020 virtual meeting, we were very happy to hear the results from Baylor College of Medicine that were presented by Dr. Spiridoula Vassiliou. They were able to manufacture trained T cells against SARS-CoV-2 from convalescent individuals with certain properties, and these cells were able to kill viral antigen-expressing targets, and this gave the indication of their selectivity and safety for clinical use. The group is now performing their clinical trial, and we are very eager to hear these results for high-risk COVID-19 patients.